You're muted, Mr. Balabhadra, you're muted.
Sorry, everyone. So, good evening, everyone, and welcome to the another session of Brains on Wednesday. For today's session, the topic is early brain development. It is fascinating, fundamental, and foundational, which, frankly, is a part of the life where neurons reproduce faster than academic theories and slower than medical equations. Early brain development isn't just about neurons multiplying, it's about how chaos organizes itself, forgets that it was chaos, and calls itself personality. So, if you ever wondered how your hippocampus learned guilt or why toddlers throw tantrums like miniature philosophers, this is your evening. It's an area that bridges neuroscience, child psychiatry, and developmental psychiatry, the very roots of understanding how the human brain emerges. So today, to maintain the sanity amidst the finances, we have Dr. Aditi Chaudhary, a clinician, academician, and mentor par excellence. He has completed.
खाली कर
He has com-
हाँ आगे नहीं करी वो हाँ
she, she completed her MBBS from MGM Medical College and DNB Psychiatry from KG Somaiya Medical College. She has worked at Rajwadi Hospital for 2 years, and since 2011, she has been a consultant psychiatrist at Park Hospital, Mumbai. She has served, she also served as a DMP guide and examiner, shaping the next generation of psychiatrists, while quietly testing the limits of human patience. I now invite Dr. Chaudhary to take over and to introduce our speaker for the evening. Dr. Aditi ma'am, the mic and the frontal cortex, all yours.
Yeah. Thank you, Dr. Balabhadra. Good evening, everybody. I hope I'm audible.
Yes.
Yeah, okay. So I would just like to say that understanding the early development of the brain is important and crucial for supporting the healthy brain development in the early years and preventing and remediating many of the threats that exist. And the extended maturation of the brain, which we now know occurs in the first few weeks of life to extending up to maybe early adulthood, it derives not only from the complexity of the process of the neurobiological growth, but also due to the brain's continuous incorporation of experience. And in order to understand this complex topic in a better way, we are very fortunate to have with us our speaker, Dr. Malay Dave, and I'm extremely privileged to be introducing him today. Dr. Malay Dave sir has, as we all know, he's a consultant psychiatrist in private practice, and he also has 12 years of experience as a PG teacher. He has been a winner of the Anil V. Shah Best Paper Award at the annual conference of the IPS West Zone branch in 1998, and also the winner of the BPS Silver Jubilee Prize at ANSIPS Bhubaneswar in 1999. He has more than 12 publications in very renowned journals, and he has served very important offices like the past president of BPS for the years 2016 and 17. He has also been a member of the IPS UG Educational Subcommittee for the years 23-24, an advisor of the IPS Biological Psychiatry Subcommittee for the years 2025 and 26, which is ongoing. His areas of interest are neuroscience and genetics. So I can think of no better person to educate us on this topic than Dr. Malay Dave sir. So over to you, sir.
Thank you, Dr. Aditi, for the kind words, and welcome everyone. Good evening, everyone, and we have another evening on of Brain on Wednesdays, and we have been the last quite a few months, I think 6 or 7 months, we have been talking about certain basic processes, and we have kind of had a snapshot of what these various processes are, what these various systems are. What these various physiological patterns of working are. So what I thought was that let us try and keep those things in mind and see if we can understand brain development better. Now, as Dr. Aditi has mentioned, it's not a process which has actually got an end. There is always a beginning, but for brain development there is no end. because certain things keep on happening, and we need to take this into account. So without much ado, I'll start my presentation. Is it visible and am I audible?
You are audible and the presentation is also visible. Yeah.
Okay,
Perfect.
right. Okay, so now in the next, let's say about 45 minutes or so, I'll try and give a kind of a panoramic view of what do we need to understand as psychiatrists about brain development. It is a very fascinating area of neuroscience, both at a basic level, both at an experimental level, and both at a clinical application level. And as like any other branch of science where we have newer technological modalities coming in, newer way of kind of interpreting our scientific findings. So also with brain development. So a lot of things keep on coming up. There are a lot of actually players in this field of science. It's not only limited to doctors, neurologists, psychiatrists, but even basic scientists who are working in the field of biology. We have uh, technological people coming in terms of people who are well versed with let's say computer programming, because we are at the stage where we can develop models and try and understand how a particular model is going to behave in terms of over a period of time, and so on and so forth. So okay, now this is what is actually the most important thing for all of us, and this is what we need to keep in mind when we are dealing with patients. So when we talk of brain development, it is actually not a line. It is not a linear kind of a thing. It has got a lot of players, and it has got a lot of this interconnecting arrows, which makes it quite complex. We can start from anywhere. Let's say we start with brain activity. Brain activity has an effect on hormones. It has an effect. It's an interacting effect with psychological and physical experience, which can have an effect on gene expression and vice versa. So we can take any parameter or any point of interest, and well, you can come back to it in a very convoluted manner. But this is what brain development is. The first thing that we need to understand is brain development is not a linear process. Second thing we need to understand is that all the parameters here, they are not hard and fast. So depending on individual, depending on a particular race, everything will keep on changing. And also everybody's experiences are not the same. So again, that has got an added input on development. So this is what actual brain development is. Now, we are all familiar with this kind of diagrams because this is what we have learned right from our very first year of MBBS in embryology textbooks. But what is important is that brain development or the development of the nervous system starts on very early, 25 days. So it's not even a month. Now, what is important here clinically is that even before a woman realizes that she has missed her period, that she is pregnant, even before she comes to know of that, there is always uh, the brain, that is the nervous system, that is developing. So around this time, 35 days or 40 days, when the period is missed more than a week or 10 days, then the lady will come to know, the woman will come to know that, she is pregnant. And by that time, there is definitely going to be quite some organized central nervous system development. And of course, it progresses over time. So, let's say this is the first 100 days, 3 trimesters, the second trimester, and birth and after birth. So around the first 4 weeks of gestation, there is the separation of prosencephalon, the mesencephalon, and the rhombencephalon. So basically, the 3 main areas of the nervous system, they get differentiated. At around 6 weeks, this prosencephalon, that is the anterior most part of the nervous system, developing nervous system, uh, divides into diencephalon and telencephalon, and rhombencephalon, that's the end part, is divided into metencephalon and myelencephalon. So this is the first trimester. Second trimester, that is the later stages, we have cerebral cortex, which forms from the telencephalon, subcortex from the diencephalon, and cerebellum and brainstem structures ventricles from this. So at 1 month or even less than 1 month of gestation, we have got distinct areas which are kind of going to develop into specific parts of the brain. So this is the forebrain, let's say midbrain, this is the end brain or the brainstem. So prosencephalon will develop into the cortex and the subcortical structures. The mesencephalon will be the midbrain structures, and this rhombencephalon will be the end structures. So this will be the cerebellum, the cerebellar peduncles, ventricles, then the spinal cord, and so on. So this is how it would be. At around 4 weeks, it will look like this. It's not colorful. This is just a legend. But yes, prosencephalon, mesencephalon, rhombencephalon, and then there's a further. So first trimester is this. Second trimester, it's more defined into individual parts or systems. And at birth, more or less the whole structure of the nervous system is in place. Now, so that's one part. So that's the gross part of the nervous system, the central nervous system. What is important is how the nervous system cells develop. I am using the term nervous system cells because there are many kinds of cells. It's not only the neurons that are important, but even the different types of glia and different types of other cells are also important. So during corticogenesis in utero, the marginal zone forms into different ventricular zones on the cortical plate. So this is later on. This is second trimester and later on. And during the same stage, we get the typical human cerebral cortex, which is 6-layered from an inside out fashion. Means the cells start kind of dividing and proliferating and migrating from inside to outwards. So the topmost cells of the cerebral cortex are the youngest. And as we move from the top of the cortex down towards the ventricles, we have got cells which are older in nature, or rather older in development. And precursor cells, also originating from the ventricular and the inner outer subventricular zones, will radiate to form layer 6. Okay, so let's see if we can understand this better. So what happens is that to start with, we have the marginal zone and the neuroepithelium. They are similar cells. So this is what we call as a, we can roughly call as something like a pluripotent cell, similar to a stem cell, which will or a progenitor cell, which will over time develop into different kinds of cells. These cells will then go to their different positions, and then we have got an organized cellular organization of the cerebral cortex, various lobes of the brain. So from the marginal zone, we have this cortical plate, okay, and this is basically the neuroepithelium. This is the ventricular zone. So these are the different zones. We need not get into this detail, but what is important is that this is how cells develop. So first we looked at how the different areas of the central nervous system develop. This is a rough diagram of how various different types of cellular, different components of the cellular part of the brain or the nervous system develops. Different kinds of cells. We have the 6-layered cortex here. Okay, and then we have other different cells, which are the supporting cells, the glial cells, and so on and so forth. Now, what is actually most important for us clinically is this diagram. Now, this diagram appeared about 25 years back in a magazine or a journal called American Psychologist. This is January 2001. So that's how we didn't have very fancy AI kind of diagrams and very colorful kind of media. So it is all black and white. But somehow, whatever we have understood in the last more than 2 decades, everything more or less fits into this pattern. So this is from conception to death. So it is how many numbers of years I have lived plus the 9 months in utero. So here we have seen that 18 to 24 prenatal days, so it is just less than a month, the neurulation or the beginning of the nervous system development starts. So we have those three parts: the prosencephalon, the metencephalon, and the rhombencephalon. So this is the beginning. Then the cells, the second diagram that we saw, this one, is somewhere to 6 to 24 prenatal weeks. So by the time a child is born, all the components are in place, but that doesn't mean that the functioning or the complete functioning is in place. Components are in place, and then they are going to undergo changes over time, which we call as the brain development. The development starts here, but what we are going to observe right from day one of life is from here. Now, what happens is, if there is a huge problem, let's say there's a genetic syndrome, and there are a lot of problems here or here, the pregnancy will not progress. It will be incompatible with life, so it will either undergo a spontaneous abortion or some other complication. But when a child is born, whether premature or assisted delivery, normal delivery, it means that more or less the components are in place. So the second challenge is how this is going to progress. So we have different waves, or rather we have got different kind of curves here. The first curve is sensory, seeing, hearing. The second is receptive language and speech. And the third is higher cognitive functions. Okay. So these are the curves of development. If you look at these lines, straight lines, these are the lines of myelination. So it's the cells that are developing. These are the fibers that are developing and which get myelinated. But what is also important, and very, very important, is that these two parallel lines, these two lines at the top, they do not have a kind of a curve. They are kind of going straight. So what is going straight on right from here, from birth, means since the components of the nervous system are in place, till the end of my life, I am going to have some neurogenesis in the hippocampus. So hippocampus is one place where there can be, rather there will be, new neurons that will be born. Meaning, in other areas of the brain and the nervous system, if some cells are dying or are dead, they will not be replaced. This specifically refers to the neurons. It may not refer to the other supporting cellular structure or the cellular components. That is one. And second is that whenever we are talking of synapses, so synapses will come when the gray matter is in place. We have got good white matter, so the myelination is adequate, then the synapses will be good. But again, that is not a static feature. It will also keep on changing depending on the kind of experiences that we are exposed to or the kind of experiences that we seek out. Things will happen without our knowledge or without our doing anything. And there'll be things that we like to do. We like to learn. We like to experience new things in life. So that is going to have an impact on whatever synapses are there. They will get modified, new synapses will form, the ones that are not used will go away, and so on. So we remember this diagram because this will keep coming again and again in different ways. So if you are bored right now and you want to switch off, then already the message has been conveyed to you. The message is this: number one, that development is not a linear process. It is actually going to move in circles and keep moving on and on and on. So there is no end to it. Something or the other will keep on happening. So this is the first take-home message. And this is the second take-home message, which is a little bit more kind of spread out in terms of components. This is a better, less clustered, less cluttered, and more kind of simple way of looking at what we saw in the previous diagram. So it's the same thing. Neurulation around 4 weeks. Neurogenesis end almost by 7 months. Neuronal migration. So nerve cells are born. They proliferate. They migrate. Now, once this is in place, there can be an excess number of neurons, or there can be cells which are, exposed to wear and tear. They have no use. They'll be killed. That is apoptosis. So lots and lots and lots, much more than what is needed in terms of neurons and supporting cells are born. They will go to their final place of function in the brain. But since there are so many of them, sometimes you need to kind of tell them that, okay, enough is enough. We have more than what is necessary, so you better now get lost. So that process of apoptosis, this is programmed cell death. So we are going to kill the cells. I mean, not we. It's a process which happens. It's a programmed process which goes on, let's say, till the beginning of adolescence. And that also is also responsible for synaptogenesis. So synapses will keep on forming. So at birth, let's say till here, we have a complete machinery in place, and there are some things which are getting extended till adolescence, before adolescence, till adolescence, adulthood, and beyond. So this is how the development happens, and this is how our brain functions. So what are the determinants? How this whole how does this show go on? So first of all, it is always going to be genetics, because all these things which are very complicated, right from how the pattern of neurulation, means how those 3 parts of the nerve axis get demarcated, how are the nerve cells going to be produced from the non-specific general mass of cells, how the neurons are going to migrate, how the cells are going to migrate to their place where they are going to be finally be present and work, and these other processes which will maintain this. It is a very complex thing. So it is not going to be just a random something happening. It is a kind of a genetic program that is laid down. And I am not going to mention the genes that are important, because there are thousands and thousands of them. In fact, there are so many common genes between various species of humans and hominids, so our ape cousins, that many of those are overlapping, and there are certain different developmental genes which make us more human. So there are literally thousands and thousands for each and every kind of a process that is involved in brain development. We like to believe that a newborn has got no experience, but we now understand that even in utero, there is a lot of programming that happens when it comes to brain development. We'll have a look at it later on. So it is both nature, and if we would like to call it nurture. So none of us are actually blank slates when we are born. There is a certain programming which we are going to see later, and why and how that happens. Both genes and environment make significant contributions to the development, because first 9 months it's in utero environment, and from day 1 of life till day end of life, it's going to be how the environment influences an individual, how the individual tries to cope with, manage with the environment, and what are the residual effects, good or bad, on the individual. There are also epigenetic processes that are important, which can come from the previous generation, that is the parents. And over a period of time, there can also be new epigenetic changes in the individual. And whatever the genome is, so whatever the genes are and whatever the epigenetic changes are, they will constantly act or interact with the environment. Now, whenever something happens like this, environmental interaction with an environmental component, there has to be some learning that should happen. Otherwise, it's going to be the same problem all over again and again, or it will be the same pattern of behavior or same pattern of a response or a same pattern of a side effect that will keep on happening again and again. And that happens because of plasticity. What are the other components? One is chronobiology and periodicity. So nature has given us a kind of a time frame in which certain things happen. We have got the easiest one to understand is the circadian rhythmicity. When a girl attains menarche, then there are the hormonal cycles which are about a month. So, and there are a lot of those internal rhythms which work on, let's say, a different time scale, microseconds to seconds and so on. So there is an element of this periodicity which determines cellular function. And of course, there can be a lot of individual variations. So if we look at the first diagram, when we look at development, it's not only static, means it's there and it remains. It is not linear and it is not universal. So there are going to be individual variations. So what are the patterns? Pattern means how does the brain develop? So one is there is differential growth, intra-regional. So let's say if we look at the frontal lobe, it is a region of the brain. Within my right frontal lobe, all the parts, all the components, all the areas will not develop at the same time at the same speed. Then there is an interregional. So again, there might be some different patterns of development in my right and my left frontal lobes. Hemisphere, even the hemispheres overall will have slightly different patterns of growth. The growth doesn't stop. So, nothing great happens when I turn 18 or anybody turns 18. It is just a kind of an arbitrary figure that people have agreed on across the globe, that 18 means after 18, we become adults. So we are given the right to vote, or we can get a license to drive a car, or in cases of girls, they can decide to get married. But we need to understand this growth and development of the brain proceeds very well into adulthood, which can actually go up to not only young adulthood, but let's say middle adulthood. There are critical areas, there are critical time periods where a lot of this pruning happens. Pruning means we talked about this part, apoptosis and synaptogenesis. So whenever we have lots and lots of cells, we are going to have lots and lots of connections. This the peculiarity, or rather the quality of neurons is they are not cells that function or they live in isolation. They are always going to form connections or synapses. So if there are lots and lots of synapses, all of them may not be needed over time. So those which are used often, those that have got important connectivity with a lot of other parts of the brain, those which require interaction with certain specific kinds of environment, they will stay, and the other synapses will be kind of censored or cut off, which is called as pruning. So this pruning is based on use it or lose it principle. So if there are certain synapses, as I mentioned, which are being used more often, then they are likely to stay. Now again, how does the brain function? So as I said, we are born with the machinery. How it works is, again, what the way it was working at birth is not the same as let's say it's not going to be the same pattern of working when I'm 18, 35, 45, 50, 70, 80, 100 and so on. So it depends on networks. Now this is the first basic science topic that we covered, which was connectomics of the brain. It is only after we understood connectomics, we understood how actually the brain functions. We got an idea of brain function more accurately when we talked of networks. So that is the nodes and the networks. And why so? Because centuries ago, when we actually had some kind of dissection and we looked at the brain, the most we could identify was the different areas of the brain. Now, they are not actually anatomically delineated. It is for our convenience that we mark certain areas as frontal, temporal, parietal, right? We say hippocampus is in the medial part of the temporal lobes and so on. But these parts and these structures, they are not isolated organs. They are a part of the same system. So for the convenience of understanding something better, we looked at parts, and then we had those Broadman areas and all. Once we had those electrophysiological studies, we would give, let's say in animal studies, we would give a stimulus and see what happens to that organism's brain. So we understood that some things happen. We give them a light stimulus, then something happens in the cortica, in the occipital lobes. We give a sound stimulus, something happens in the temporal gyri, and so on and so forth. Now that gave us a belief that there are different areas of the brain which are distinct lobes, temporal, parietal, occipital, this thing, that thing. And these lobes have got certain functions. But the more we understood networks, we understood that it is not like that. For a certain function to be carried out, it is going to be an integrated effort of multiple areas of the brain from multiple lobes. So what is more important for us to understand is how the networks function rather than what the lobes are doing. Because when we study lobes from the neurology perspective or from older textbooks, it will always start with functions of the frontal lobe. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Functions of the parietal lobe. But the point is that those lobes or those places will be one component of an entire network which subserves that function. So unless that nodes and networks are in place, the brain is not going to function optimally. Now, once this kind of a functioning is set, then there is something called as lateralization. Very kind of a how would I put it? A very kind of um, unusually simplistic way of putting that I have got a right brain and a left brain. But where it stems from the fact is that the networks that are present in the left hemisphere may be more efficient in processing some kind of stimuli or some kind of information compared to the right. But it is not always true. If there is a Broca's area on the left side, doesn't mean there is no Broca's area. It's only that all kind of researchers focused on that Broca's area on the left. The right will also be doing something similar, but not to that extent. So there is something called as lateralization. Now, why do we have this kind of organization and lateralization? So that the brain can work very efficiently. If in an organization, I am going to do everything. I am going to do the CEO's work, and I'm also going to do the janitor's work, and I'm also going to do the cleaning work and this thing, that thing. That organization is actually going to be very, very difficult to sustain, because there is, everything is centralized. Now, this is going to take a tremendous toll on the resources, not only resources, but on the processing efficiency. So everything that I need to do is going to take a lot of time if I function that way. So it is organized. It is decentralized. And even by decentralization, it is again specialized. So everything runs very smoothly. Everything runs in an integrated manner. And everything actually, because it is working well, the central commanding authority will have no problems in understanding what is happening and intervene when needed. So it runs very smoothly. So these three things, synaptic pruning, formation of networks or connectomics, and lateralization or specialization of function makes the brain function very efficient. And these things, they evolve over time. And to add to that, sorry, there is always the sexual differentiation. So XY, XX. Now this gives a this gives a differential response of brain cells to different hormones. The effect of testosterone on brain function, the effect of estrogen on brain function are slightly different. And that is where we call that, we call this as sexual differentiation. After menopause, when the influence of the female sex hormone is less, then the brain function starts becoming more or less similar, but not completely same. So what happens? Now, when we look at this graph, it is actually too much of information. But what we need to understand here is this, that this is a part of the cerebral cortex. This is part which is a square with white matter underlying. So this is the magnified part which is being studied. The green is the surface area of that part. The red band is the cortical thickness. This is the volume of the gray matter. This is all the gray matter. Okay. And this is the white matter. So what we are looking at, we are looking at the gray matter, we are looking at the white matter, we are looking at the surface area and cortical thickness. Now, as I mentioned, this is again from the post-conception period throughout life. Again, the brain volume. If you look at brain volume, we look at these four things. We look at gray matter volume, white matter volume, cortical thickness, and surface area. They are not again same. They keep on changing with the cortical thickness, which becomes maximum, let's say around birth, and then it kind of remains. And as we age, the cortical thickness becomes less. So this is still around 70, 80. So if we take our MRI scans, if I have an MRI scan at birth, and if I take it 80, definitely the cortical thickness would have reduced. If we look at the volumes, the gray matter volume and white matter. So this light blue is here. This light blue reaches a peak, gray matter, at around 6 years, and then starts coming down. And if you see, this gray matter has got a U-shaped thing. It's an inverted U-shaped curve. Compared to white matter volume, which is this one, which is more or less like a gentle curve, or the surface area. So this is the area that is what we are looking. We are looking at the different parameters and how just in terms of sizes, whether it is area or volume, how the brain behaves over time. To put it in very simple words, this is the first thing that I mentioned here. Differential growth. And what is this differential growth? Frontal lobes, temporal lobes, parietal lobes, the three main lobes, they have their peak volume, peak thickness in girls at 11 years, boys at 12. Now, if you see, for the girls, it is slightly there, apart from the temporal lobes, they achieve this volume earlier than the boys, except temporal lobes. So, in boys and girls, we say, this is again what we know from what we learned, is that girls tend to mature earlier than boys. There is a gap. If you look at the frontal lobe development, that is a gap of about a year. But there might be some problems, but not problems, some lag of emotional development by about 6 months, wherein girls lag behind. But when it comes to sensory processing, they are 1.5 years ahead. So again, different lobes, both in boys and girls, they've got a different way, or rather a different speed of development. So we talked of cortical changes, inverted U pattern of growth. gray matter increases till onset of puberty and then begins to become thinner. This is because the excess of synapses are getting pruned. This is again dependent on experience. So people, the children who are, they are socially deprived, they are nutritionally deprived, okay, or they face abuse, then this cortical thinning will be faster. How does this maturation happen? So this is those three curves that we looked here. This is the pattern everywhere: sensory, speech and motor, and cognition. So the first thing that is going to mature is the sensory system. The sensory systems have to be in place because anything and everything that the brain is going to do is dependent on the kind of information that and the quality of information that it gets. So the sensory systems have to be super class. Once that is there in place, then the behaviors, what to respond will develop. So the motor component, but the connection between the sensory and the motor will keep on improving. So I get angry, I'm not happy with something, I throw it away. So sensory has developed because I know now this I don't like. Motor, throw it away. But as the cognitive processes improve over time, then I'll know that it might not look okay or taste okay or feel okay, but it might not be bad. So this is the cognitive development which happens last. So the maturity is sensory, motor, and cognitive. Now we looked at the cerebral cortex, but again, we did not really get into all the minutiae of this diagram. But what does it show? It shows, if you look at from A to L, different parts of the brain. So A is thalamus, B is cortex, C is cerebral white matter, D is cerebellum gray matter, and so on. So all the parts of the brain are labeled from A to L with this color. And when we look at the gray matter thickness, we see that again, the gray matter in all these things is not the gray matter thickness or the volume is not it doesn't follow the same pattern everywhere. All the curves are differently placed. So again, it's not only about cerebral cortex, but what happens at a subcortical level, what happens at a brainstem level is also different from each other. White matter. White matter is important because all the information that the gray matter has to get and all the information that has to be relayed from the gray matter downwards to all the effector systems, it has to be smooth. The roads have to be smooth. We cannot have potholed roads. When we have potholed roads, all the information gets haywire. It doesn't get transmitted well. So this smoothing of roads is called as myelination. And again, like the gray matter, this myelination also is a little bit not uniform. It happens, it is achieved early on in certain parts, and it takes a long time in certain other parts to be fully myelinated. So bilateral strengthening of the connections in the superior longitudinal fasciculus. So superior longitudinal fasciculus is the long one from one end to the other of the brain, anterior to posterior. But here in white matter also, the myelination happens in the same way. First is sensation and movement, and then all the other cognitive functions. So again, it's the same thing. Myelination also increases and then kind of reduces, influenced by experience, and it extends well into adulthood. So I decide to vote for somebody at 18 years of age. When I become older, when my myelination is good, when my brain, when my gray matter is more evolved, well connected, my networks are in place, then I might think that no, that was not the right thing to do. Or I liked somebody at 18 years of age and I want to kind of run away with her and get married. And then after 10 years down the line at 25, 30, I might really not be happy with that choice. So this is what happens. The development of both the gray and white matter proceeds very well to late 30s or early 40s. So this is the white matter. So association tracts, superior longitudinal fasciculus, these are mature by adolescence. So long-term message relay is okay, grossly fine, but the smaller ones, they are still developing and immature during adolescence. So this leads to, as the white matter becomes more evolved, this is refinement of networks. So in younger children, the brain processes in a more locally distributed. So parts of the brain will kind of process, but more focused and efficient. So integrating all takes time. So as long range connections increase and specialized brain networks develop, the workload of cognitive processing becomes widely distributed and integrated. And this is the basis of all the networks that we have studied. So whether this is the default mode network. So again, at birth and in children, all the components are in place, means the system is there. But as we become older, the connectivity, if you see, it's like a puzzle. You know that puzzle where the question is asked, is the number of the red dots same? Actually, it is the same. What is differing in the two is the black lines. And how thick are those black lines? The black lines indicate the edges or the connection between these nodes. So reds are the same. The shape looks different only because of the blacks. And this is because of the myelination that happens, the connectivity that happens as the network matures into adulthood. Same thing happens with salience network. So that was default mode network. This is salience network, but the same thing. So again, this is a more of an fMRI picture, and we can see that some connectivity is different between adults and children. Similarly, if we look at a more brainstem function, which is the eye movement function, and how that connectivity changes our behavior. So in a newborn at 1 month, 2 months, and 3 months, the connectivity keeps on improving. Only suprachiasmatic with layer 4 and 5, plus inhibitory, via basal ganglia, and all those. Now, initially it was only tracking movement. So an infant can look here and there, may not be able to track well. But as it becomes older, it can track and actually keep the focus on that. Okay, that smooth pursuit becomes more better, and 3 months and above, they can actually anticipate where something is going to move. So it is not only the motor component of the eyes that is important, but the whole system of connectivity between the eye movement and what the behavior is going to be also improves. Again, all these structures are in place. But when the inter-regional myelination improves, then this process progresses this way. Another thing that happens is sleep, because again, sleep is not something which is a miracle or something which is, just fancy random. It is a multi-component output of various brain centers. So children and adolescents have a delayed sleep phase, means they're going to sleep late and wake up late. Most of them will have an evening chronotype, means they're more active in the afternoons and the evenings. So many children, they'll complain they'll not wake up on time for school. They can complain of tiredness and boredom in school, which should not be actually diagnosed as anything. It is because their physiological sleep architecture is that way. But an almost adult sleep pattern will take time. Okay. And again, there are lots of areas that are involved: locus coeruleus, suprachiasmatic nucleus, and so on and so forth. So this is how it will move. At newborn, there is many hours of sleep, 16 out of 24, half NREM, REM. As we become older and as we become very old, what happens is we'll have more of NREM sleep, less of REM sleep, but total number of hours of sleep is also reduced. So this is the structure. We'll see there's a curve here. So this is the curve of the slow wave sleep. At 6, it is going to be maximum 6 months to a year, 6 months to 2 or 3 years, and then it will come down again. But all the components are less. So as we become older, so this will be a lot of dreams. Here, fewer dreams, fewer realistic dreams. Sleep can get broken easily, and so on. Okay, now this is basically the last component of the development. So we looked at the gray matter, we looked at the white matter, we looked at the networks, we looked at sleep. This is the central executive, so which is going to subserve all cognitive functions that we put in those charts which tell us the development of the child. So conceptual structures emerge from basic sensory motor skills. So sensory and motor have to develop first. Then there is higher cognitive system involvement because there is a pressure for information processing. Working memory span starts developing at 6 months, spatial orientation, 5 months, this we saw. The oculomotor development. Infants can adjust their grasp and reaching for objects in the dark. These are just how, this is again not a complete list, but this is how things will move. Infants can rotate objects to fit them through holes. 22 months, this takes time because it is the sensation, it is the motor activity and the cognitive processes, the hand, eye, the oculomotor development and so on. Active mental rotation. Now this is important because at this point, the child is able to hold on to an image and kind of figure out that if in a puzzle, I'm going to put it this way, it may not fit. So they become better at puzzles. Okay. Motor competency and so on and so forth. So now all these are cognitively mediated outputs. So there is a sensory pressure, there is a motor output, and something is happening by which the motor activity becomes very precise. And this is what are the areas for reaching, for grasping, spatial attention, working memory, and so on. So they are there. Again, I'm telling for the nth time that these things are there. Only how well their connectivity becomes over time, whether there is necessary pruning that has happened, then it will work better. Finer things, emotionality, risk-taking, impulsivity. So other areas of the brain like nucleus accumbens, which is a reward center. So adolescents will show increased ventral striatal activity. So they are more dopaminergic, let's put it that way. And the peak volumes of this are reached around adolescents. So they are going to be extremely impulsive. They can be they will get into, I don't like this, get it out of my sight. They'll rebel, they'll experiment. Okay. Now, left nucleus accumbens in females have smaller, but males tend to have larger left nuclei. So there is a gender difference. We talked about those sex differentiation. This is something. So as the puberty progresses, females are smaller, but males tend to have larger left nucleus accumbens. Amygdala. Now, amygdala is something which is one structure of the brain, which is there in its complete form from day one of birth, not conception, but birth. Because any threatening stimulus or fear-inducing stimulus has to be picked up much more faster than any other stimulus. So that system is in place. The only thing is how it is going to be interpreted, how our memories are going to be encoded with a fear stamp, whether it is actually fear or not. How do I manage my phobia? That is going to take lots and lots of many years because the connectivity of amygdala with hippocampus, with nucleus accumbens, with prefrontal cortex, that will improve over time. So amygdala volumes increase in both boys and girls, but hippocampal volume decreases in girls as puberty progresses. These are findings. These have been found in repeated studies. Now, are the associated findings, so they are always true in everybody? Is always going to be a matter of debate. So now what makes us quote unquote mature human beings? It's the prefrontal cortex, which has an inhibitory role over everything. So one of the most important cognitive function is inhibition. Inhibition is a general thing, but anything which requires kind of a censoring of the output, modulating the output, modulating the impulse, modulating the reward systems is inhibition. Okay. Why does it become better? Now, when we looked at the cortex is 6-layered. So there are 6 layers of cells. It is called the neocortex because in human beings it is 6-layered. In other smaller rather older creatures or smaller animals, it is less layered. So what's the big fun in that? When we have 6 layers of cortex, this cells between layers will also communicate. So the organization of the cortex is such that one component, one column of cells across the 6 layers will form a unit. And then there are millions and millions of such units. Now, each unit is like a, if anybody has kind of opened a phone or a circuit board, there are so many circuits, and there are those small kind of capacitors or transistors which will, uh, uh, you know, multiply the signal or reduce the signal. Okay, we have those kind of networks in the cortex. So this network, each network is connected to the other. So any output from the cerebral cortex which is going down will pass through all these things. And the inhibition is GABA. This GABAergic activity is extremely complicated because there are different types of GABAergic neurons. We tend to put everything under the GABA interneuron, but there are 3 or 4 different subtypes of GABA interneurons. They will fire at different points in time for different types of impulses, for different types of inputs. So certain circuits will get inhibited, certain will not get inhibited. Now this fine tuning again will develop on how well the cortical cells are in place, how well they are connected to each other, and how the whole cortex is connected downstream through the white matter. So this takes the most time to develop. Point is, if I tell my teenager that what you're doing is not right, may not be okay, do this, it's not going to have much of meaning or a long lasting meaning, because even though he or she understands, the system of inhibition of things is not fully developed or in place. That brings us to this dopamine thing, because the previous, or rather the talk here is about impulsivity. This is an example that when we are looking at reward systems, and if we look at one of the components that is dopamine, other neurotransmitters also, but dopamine, because all of us are familiar with dopamine more than anything else, is that dopamine doesn't have, again, a stable functioning. There is a tonic dopamine activity, means there is some amount of dopaminergic activity going on, because if this was not there, I would become apathetic. I will not respond to any stimulus in the environment, internal environment or external environment. And then there is a phasic activity. So there is a peak. So there's a baseline activity, there's a peak activity. Now, this is an adult form of dopaminergic activity. With time, with maturity, the activity is modulated so that unnecessary peaks are not there. The inhibition from the prefrontal cortex is good. So lot of impulsivity that was there are many people who will tell us that, I was a very outgoing person, I was a very naughty person, I used to be very bindas, but over time now I become subdued. So that part was also not abnormal. This is also not abnormal. But this is how the organization of a neurotransmitter system of dopamine has happened over a period of time. It is now optimally used with proper inhibition from the prefrontal cortex. From a human perspective, these things, when they are in place, we kind of label this as the social brain. So decision making is mature, regulated, there is impulse control, risk behaviors are less, emotionality is modulated, self-regulation is good. I am able to, we say, I have the theory of mind, I can empathize. But all these higher order social functions, they just do not happen out of thin air or just by teaching somebody. What is going to work is, if we look at it from what we have learned before, these are outputs, these are higher cognitive outputs, these are complex cognitive outputs. This will happen only when the brain development is proper. When we have talked of those other things, all those things they've happened in a required normal spectrum, and they're in place. Otherwise, these things will not work. So what happens is that in an adolescent, well, this is the brain when we are looking at the brain from the bottom. So this is the cerebellum brainstem. These are the prefrontal cortices. This is the orbitofrontal cortex, and this is a lateral prefrontal cortex or dorsolateral prefrontal cortex. In an adolescent, the processing and the analysis happens here because a lot of emotional input is there. The orbitofrontal cortices modulate emotions, analyze emotions, and that's it. But as the connectivity improves, these actually are parts of the salience network along with the default mode network. So more of control moves from here to the lateral aspects of the prefrontal cortex. This is where the decision making becomes good. Here the decisions are more random, spontaneous, emotionally decided. This is the more mature one. So very roughly, an adolescent brain is predominantly working at this level. A mature adult brain is working at this level. With an ideally developed brain, we have a detector of stimuli coming from, let's say, what we hear, what we see in terms of faces, okay, mood regulation and cognitive regulation. So this actually are parts of our mental state examination, roughly. There are too many finer details, but this is how it will work. So the whole brain with small components, small things works like this. Now, what are the parts, what are the things that will affect brain development? Though that what we looked at was normal brain development, so to say. So one is maternal antenatal stress. Okay, this is a kind of a review. And what has happened? Maternal antenatal stress and outcomes for infants and children. So infant temperament is affected, development and cognitive function is impaired, emotional and behavioral problems can be present, and there can be structural brain changes. So a patient, a woman who has got a psychiatric illness, undergoing a lot of stress during pregnancy, can be associated with a poorer outcome in infants and children. That needs to be addressed. What mediates this? Something like this is happening. How is it mediated? So major life events, catastrophic disasters, chronic stress experienced by the mother. The effects can be seen in the fetus via the means of the placenta. We'll take this topic later on because I had spoken about this in details in a different setting. But now we understand that placenta itself is just not a passive organ for nutrition, blood supply, but it also modulates, mitigates, magnifies a lot of things that can be passed to the fetus. So there can be epigenetic changes in the placenta, so many of them, because of maternal stress. This maternal stress via the placenta can also have an effect on epigenetic changes in the offspring. Certain hormones like human placental lactogen, which are secreted by the placenta, can also change the placental structure and function, right? So whenever there are certain physiological or more than physiological changes in the pregnancy, this can be the first indicators clinically. Things are mediated from the mother to the newborn or the fetus via the placenta. So during pregnancy, I mean, if there are fetal abnormalities and all, that's a different thing. But if there are certain issues regarding the placenta or the progress of pregnancy that are there, then there are variable outcomes that people have studied. So some things can be seen in the neonate and infant, some things can linger on, get into childhood, and some things can be seen in the adulthood. So this kind of a thing in a mother who has developed a depression and anxiety, which is again her vulnerability, her epigenetics, can be modulated both ways, in a good way or not a good way, and cause changes in the fetus and problematic fetal outcomes in some. Another factor is the gut-brain axis or the microbiome of the mother. So I'll just take 5 minutes more and we are done. We used to believe that the placenta is, the amniotic fluid is sterile. But what people have found out that amniotic fluid is not sterile. There is a very small amount of uh, microbiome in the placenta. There are very few species of bacteria, but they are there. They are not going to cause any problem, but maybe they are important in the neonatal development, and that's why they are there. So that is the first thing. But over time, let's say with breastfeeding or formula feeding, the microbiome is going to change. The constitution of the microbiome is different. So it is mostly aerobes to start with, and then it becomes more anaerobic. Now, why it is important is that this microbiome itself is a source of immune function or dysfunction, neurotransmitter function, good or not good, right? So if the microbiome is not healthy, then it has an additional effect on brain development, because the gut and the brain, they all develop from the ectodome, the same cellular origins, and that's why they're deeply connected with each other. So an effect on one can always have an effect on the other. This is basically the mechanism of plasticity. So this is the NMDA, and the more NMDA works, the more plastic changes will happen. Now, why do we need plastic changes? Because we need to learn. We need to learn from environment. We say it's an environment, but what actually happens is that we learn from our response to the environment. If the response is good, we remember it. If the response is bad, we also remember it. If you want to learn something new, we have to remember it, and that is the basis of learning. Plasticity is one of the things that happens at a very microscopic level. So this is something called as long-term potentiation, where there is an optimal function of the NMDA receptors. Now, why we are talking about this? This is the basis of the pruning. If I'm using this synapse, let's say I'm learning music, so I'm doing practice 5 hours a day for years together, then there is long-term potentiation. So I will learn. Then I get bored of it. Then I don't practice. Then I'll kind of not be very good as I was before. So this is called as long-term depression. Potentiation, depression, learning, forgetting. Now the basis of this is the glutamatergic mainly, NMD and AMP, how it functions. But if I am going to use this again and again, this synapse will not undergo pruning. This will remain because I'm using it. If I stop using it, there can be a point in time where I don't remember much of it at all. So there has been some synaptic change. Some of it has remained because I've used it, but not as efficient as before. So this is the basis of learning, forgetting, pruning. Now, whenever there's chronic stress, we looked at that stress, maternal stress, that was one thing, early childhood stress. Now, this causes a lot of change in glutamatergic neurotransmission, how the astrocytes interact with the neurons, glial activation. So it actually sets a tone for chronic low-grade inflammatory changes in the synaptic function. This is not going to keep the synapse healthy. This is not going to keep the system healthy or the network healthy. So when everything is in place, this is just to give an example. This is a model which all of us have seen when it comes to addictions, any kind of addictions. The addiction process is there, only what is being the source of addiction will differ, substance or behavior or food or whatever. Now, whatever be the complexity of this, so this is shown the different areas and the connections. But one, here we will have the development of, or rather the not good development of impulsive behavior or control or inhibition of that. Second, we are going to have problems with executive function because there might not be proper prefrontal development or prefrontal connectivity with other systems. Third, overactive reward systems. So there are so many components to it, but point is that when there is some problem with development at multiple areas, with all the influences that we have talked about, maternal susceptibility, maternal illness, or a family history, or early childhood adversities and stressful situations, or illnesses, then and of course that overactive glutamatergic system, there is so much that is set into place, set into motion, that there is a chronic neuroadaptation which is unhealthy. This leads to something like addictions, which is a very simplistic view, but something like this also happens in many psychiatric illnesses, maybe most of them. So what are the implications? Why do we need to understand? So basically it is one, from the physiological aspect of learning. Second is what we can do to improve executive functioning, what kind of stimulation may be needed. We looked at sleep deprivation. So there are some things which are going to be there, which are developmentally there. Strategies for self-regulation, how to, each and every small component is an implication by itself. Especially when we are dealing with youngsters, we need to understand them better, how we can optimize our brain function in brief. So that's from my side. I hand over the proceedings to Dr. Aditi. Thank you so much.
Thank you, sir. It has been a very informative lecture, and I think you have presented it in the most lucid manner with the help of very good illustrative figures. So I'm sure that the most of the participants must have really had a lot to take home after your talk. I cannot see any questions on the chat box. Does anybody have any questions? Okay. I would like to ask one question, sir. As you said that use it or lose it, and now there is an increasing trend among parents to keep on sending their children to multiple type of classes. That is, they want their child to go for a drawing class, a singing class, and then learn musical instruments, and then sports, and so many other things. Whereas the previous generation or the generation before used to just spend their free time loitering around. So, do you, after your talk, do you positively recommend that children must be engaged in such type of various activities in order to strengthen brain development, or we can leave them to their own devices like in the days past?
So the problem is this, that when you mentioned that previously, let's say one generation back, our generation, we used to loiter around. The thing was that there were so many children available to interact with.
Right.
Vacation time or free time was you go down, play, go to somebody's house and play. So there are so many children around.
Right.
So a different skill set was established at that time, which is how I deal with somebody else's behavior, how I deal with myself. Suppose I am playing something and I don't do well, how to deal with the disappointment. Suppose somebody is fighting with me, how do I fight with that person? All these are skills. Okay. Now what happens is that, or what has happened over time is that you can hardly see children playing in the ground. It's actually become a rarity. It's very uncommon or unusual these days for somebody to tell me that I go and play, I come home from school and then I go down and play for 3 hours. That doesn't happen. So the set of play or set of things that children play with has become different. That's one thing. Second thing, do I advise parents to put their children in various things? No, not at all. In fact, what will be more important is at some point the parent should understand what the child is comfortable with. So in a group when I was playing, if they were playing something which I didn't like, I would have not participated in that. And my play group would have understood that he doesn't like this. So I was automatically comfortable with something and I knew what I was not comfortable with. Now, when parents send their children everywhere, it is like a, block this thing. So everything is getting, my musical abilities are getting stimulated, my drawing abilities, but the problem is I may not want that at all.
Okay.
So the challenge is, how will a parent understand? We are talking of normal children. How well a parent is able to understand a child's ability. Okay, चलो उसको डाल दिया किधर तो, but नहीं जम रहा है तो pressure करने का कि नहीं करने का. That doesn't happen.
Okay.
So the thing is fine, put. If the child likes it, continue. But then what happens is that it doesn't only remain to the child's liking or not liking. It's because वो बच्चा उधर जाता है. Okay, that child is doing so well in that course. Why are you not good at it? So then that becomes problematic.
All right. Get it.
You're mute. You're mute. You're mute.
No,
As
I'm
a
not.
cons,
No.
no, I was going to speak.
Ah, okay.
I may ask a question. As a corollary to what Dr. Aditi was saying, the other way around, now there is a lot of sensory and all forms of deprivation with this integrated coaching, which starts very young, like say right from 9th, 10th, 11th, 12th. So what happens to the brains of children who are almost in prisons? Like you have some centers where they provide hostel facilities,
Yeah. Yeah.
and all they do in that 24 hours is some 12 to 14 hours of
Yeah.
physics, chemistry, and maths with absolutely no other stimulation. What happens to the brains? I mean, what kind of a generation you think when you said that in adolescence it's important as to how the pruning and the
Yes.
this thing happens?
So there are two or three things that we can think of in such a situation. One is that as human beings, forget where we are, what we are doing. As human beings, we have got a brain which is capable of interact with anything and everything you throw at it. It doesn't discriminate. It always takes everything as a input. That's one. Second thing is, for most people, in given any situation that is given, most people will try and adapt to that. Third thing is that all of us have got certain vulnerabilities. We can be vulnerable to something, some physical illness. We can be vulnerable to some psychiatric illness. We cannot be sure of our vulnerability unless we develop something. I might have a family history of something. My mom had this, my dad had this, my sister had this, my brother had this. Is an indicator that I am susceptible to something. What, I don't know till I develop something. And if the counter mechanisms are good enough, I might live to whatever and not have a major problem. Now, what happens is that set of situations are different. As I was talking to Dr. Aditi before, that we have grown in an era where we are on the ground. Vacation is, we are not at home at all. We are playing in the morning, afternoon somebody's house, evening we are playing again. So it's that kind of thing. We have also done our medical studies or engineering studies in the same institutes without going to the classes. So point is that in our situation or in the current situation, the challenges are same. The environment in which the challenges are being managed or worked upon are different. This set of things can trigger somebody's vulnerability, and in another situation it can trigger somebody else's different vulnerability. Means, in this integrated kind of a thing, those who are vulnerable for that kind of an environment, to that kind of environment, may have serious psychiatric problems or medical problems or severe adjustment problems. Now, when we are looking at it, what we can do, so and of course, since the environment is different, the challenges are different. So those skills which I would have developed in a different environment, I will not get a full chance to optimally develop those because there is no challenge that way. So what happens is that you see a lot of kids or a lot of children who are in this kind of environment who are, first I get to see them a lot because in the first year or something like that, in the first few months, they figure out that they're just not able to adjust. And if they are wise and the parents are wise, the child has to be out of that one, which is a good outcome, which is actually a best outcome in all this mess. There are some children who pull on because we have fought so much, we have studied hard so much, so we need to do this, I need to get into it. They may really end up in a mess because they are kind of first not able to really manage everything with whatever resources they have, and it's an added burden of as the uh, coaching progresses, the level of difficulty. So they are very likely to end up with something. So if I want to make it very simple, children now, they are exposed to a different set of circumstances compared to, let's say, our set of circumstances or our kind of environment. Certain skills require certain things. There are broad skills, and then there are certain skills which are finer for a specific environment. So this interpersonal skills, fighting skills, not necessarily to fight, but at least to be able to put forth your point, to argue out. So many things which develop in, let's say, a playground will not develop here. This is a different kind of a playground, of course, but there is no game here. So plus the different kinds of pressures and all. So environment different, set of challenges different, vulnerabilities, susceptibilities, how it will act out is different. So the point is that when we hear of all these students' suicides and all, it doesn't happen at random. We have not looked at that individual right from day one. It is assumed that somebody who is doing well will always do well. It is not so. There's no rule like that. I'm used to so much of stress, and you give me some more stress, assuming that I'm going to be able to manage that with my susceptibility, it's no rule. So unless something happens, we will not know. But yes, if we are looking at it from the social perspective, then we have to kind of see what we can do, and that is where we need to wake up, because there are all these things that are in place. When we look at development, there are so many things. How an individual manages himself, his impulses, his emotions, that becomes a problem.
Just one last question. The excessive use of the mobile phone and the internet is a very common presentation now amongst even younger children and early adolescents. Inflicts a lot of cognitive damage in itself. You had shown the model of substance use and all. So, is there any hope for these children? I mean, if they ever were to give up the internet and the excessive use of the phone or gaming, is there any hope that they might get cognitively helped, or they are kind of done with as far as their
So it's like this, that this is a generation. The first time I used a mobile phone, or I got one, was when I was around 30 years of age. Till that time, there was no mobile only, so there's no question of getting one. But my daughter, my child, was born in the era of mobile phones. So it has become an integrated part of their lives. That is one thing. Second thing is, most of us now also work with screens most of the time. Whether we want to learn something, whether we want to put in our records, we always use screens. So a generation that is born with screens, they are there in place. We are using screens. So what is going to happen is, or rather what has happened is, that all our jobs and what work we are going to do and what work our children are going to do is always going to be mostly on screens. So screens are inevitable. One. When there are screens, there is going to be every kind of thing. We are going to have the net and small screen, large screen, medium screen. Screens are there. by itself it is not a problem because it is there in the environment, and by being in the environment, by virtue of that, and by virtue of what we understand from brain development, is the brain is going to perceive it as a challenge. Any challenge is going to be processed, analyzed, worked upon till normally there is mastery over that. That is what is the point of development. What do we mean by maturity? We talked about that. So let's say very good impulse control, inhibition. So now when we are using something, we are going to be we are going to be we are developmentally actually prone to use it, to master it. That is what human beings are. That is why we have survived. So that is inevitable. So we don't kind of talk about whether screens will go. That is all fantasy. Doesn't happen. The question is then, where is the problem? The thing is that on my mobile phone, I get every week I get a message how many hours per day I'm on screens. On my mobile phone, I'm not talking of the laptop. And mobile phone, my average is around 8.5 hours a day. So what I'm doing for these 8.5 hours? Most of the time it is with patients. So whether it is messaging or talking or there's a video call, most of the time is that. Most of the time. 60-70%. Sometime is reading stuff. I don't even read things on my laptop. I read on my mobile. Whether it is papers, whether it is whatever. Now, the problem then is what is actually to be done. It is not the screen. It is going to be there. In fact, in most schools now, homework is done on screens. Projects are done on screens. So it is there. The question is the content. And at what point in time and in what manner a child is interacting with that. So when we were children, when it would get dark and we are playing throughout the day, strict warning, 6:30 p.m. you have to be at home. 7 is like too late. Why? Because it becomes dark. And when it becomes dark, we cannot wander off anywhere. Because it's a bad world. Now the bad world is on the screen. So we followed that. Now we have to follow some kind of precautions because it is wishful thinking that it is not going to be there, it will go away. In fact, when people say that, I have not exposed my child to any screens, my worry is what will happen the day when the child is exposed to the screen for the first time. And it's not going to be under your supervision. Some friend will introduce it, something here, something there, which will be more problematic. So once we accept the reality, this is going to be there, then we try to figure out what is the best way to do things. So when we try and kind of do this in a responsible, friendly way, then there's a better chance it will work. But it cannot be a group activity. It has to be each family's activity. If my mother and my father and my grandparents have told me that I have to be home at 7 and everybody agrees to that, then it will work for me. So the challenge is the content and when and how is a child introduced to that rather than the screens per se. If there was no screen, we'll not be having this talk at all.
Correct. Thank you, Davisa. And I would now like to hand over the proceedings to Dr. Balabhadra.
Thank you, sir, Dr. Dave, for a brilliant and reality check talk. My early brain clearly missed some of those critical windows, probably the attention network ones. You just have proven that that early brain development isn't only for the infants. Clearly, ours can of still grow midweek. We'll now open the floor for discussion, but I see that there are no questions. So we could just have the closing remarks now. So neuroscience rarely gives simple answers, but it gives beautiful confusions, and that's the progress. Thank you, everyone, for joining tonight's Brains on Wednesday. We have learned that the brain starts shaping itself long before we start shaping our careers. And thankfully, neuroplasticity allows both to recover from the early mistakes. As we wrap up, one key takeaway stands out. The brain remembers how it was nurtured. What begins as biology becomes biography. Thank you, Dr. Malay, for the clarity, Dr. Aditi for the structure, and the Alchem CNS funding for collective obsession within our system. To all our participants, thank you for being part of this growing community of leaders tonight. We'll see you again on the next Brains on Wednesday, same time, same neurons, just a bit more evolved. Don't forget, we meet every second and fourth of Wednesday at 8 p.m.
On
I'm
behalf
Dr.
of
Balbhadra Singh
BPS
Jadeja.
also, thanks a lot. On behalf of BPS also. Thank
Yes,
you, Dr.
sorry,
Malay.
ma'am.
Thank you, Dr. Aditi.
Thank you, Bombay Psychiatric Society.
Yeah,
Yeah,
thank
sorry,
you.
ma'am, I missed that. Just
Okay.
had second time.
Yeah.
Have a calm evening and an active rain. Good night.
Bye. Good night.